Good morning, students. This is 11th standard botany video class, video 5. In this video, we are going to continue the lesson respiration. Dear students, in the previous video class, we have seen aerobic respiration and its different stages, that is glycolysis, link reaction, Krebs cycle, and electron transport chain. Now we are going to see the another type of respiration, that is anaerobic respiration or fermentation, in this video. Dear students, see the topic anaerobic respiration or fermentation. Some of the organisms doing the respiratory process in the absence of oxygen. That process is called fermentation or anaerobic respiration. Anaerobic, that means absence of A, means absence of aerobic respiration. That means absence of oxygen. Some organism, they are doing the respiratory process is known as anaerobic respiration or fermentation. This is divided into three types. One is alcoholic fermentation, lactic acid fermentation, and mixed acid fermentation. First one, alcoholic fermentation. In the root region of a plant which contains cells which are absorbed large amount of water and mineral salts. So these roots which are respire by alcoholic fermentation because they lack of oxygen, that means absence of oxygen, by converting pyruvic acid into ethyl alcohol and carbon dioxide. That means roots are present in the soil. Under the soil, water is also storing there. So these root cells respire by alcoholic fermentation due to the absence of oxygen. The pyruvic acid converted into ethyl alcohol and carbon dioxide. Some of the lower organisms such as yeast is a unicellular fungi. Binomial name is Saccharomyces cerevisiae. Respire anaerobically, that means in the absence of oxygen. So this process is taken place in two steps. First one, pyruvic acid. The formula is CH3, CO, COOH, 2 molecules converted into acetaldehyde, that is 2 molecules of acetaldehyde and 2 molecules of carbon dioxide liberated. Then the second step, this acetaldehyde is get reduced in the presence of NADH and hydrogen, converted into ethyl alcohol and NAD by the enzyme alcohol dehydrogenase. First step is taken place in the presence of the enzyme, that is pyruvate decarboxylase. So these are all the two steps which is taken place in the alcoholic fermentation. So the main theme is pyruvic acid is converted into ethyl alcohol and carbon dioxide. It is taken place in two steps. First step, 2 molecules of pyruvic acid converted into 2 molecules of acetaldehyde and carbon dioxide. Then this acetaldehyde reduction process is taken place in the presence of coenzyme NADH2, converted into ethyl alcohol and NAD. Dear students, important 5-mark question. Difference between alcoholic fermentation and lactic acid fermentation. First one, carbon dioxide is released from pyruvic acid and also it produces alcohol. In lactic acid fermentation, it produces lactic acid and does not release carbon dioxide from pyruvic acid. Alcoholic fermentation taken place in 2 steps, but this one taken place in a single step. It involves with the 2 enzymes pyruvate decarboxylase with the magnesium and alcohol dehydrogenase. It uses one enzyme, that is lactate dehydrogenase with the zinc. Then it forms acetaldehyde as intermediate compound. Here there is no intermediate compound is formed. It is commonly occur in yeast, but this one occur in the bacteria, some fungi and vertebrate mussels. That is the difference between alcoholic fermentation and lactic acid fermentation. Next we are going to see important 3-mark question, industrial uses in alcoholic fermentation. First one, this fermentation process used in bakeries to prepare bread, cakes and biscuits. Then it will be used in the beverage industries, that is alcohol preparation, wine, beer, brandy preparation industries, that is alcoholic drinks. Then it produces the vinegar, that is acetic acid, which is used in the tanning and curing of leather. So in leather process, the hard material of the leather product is converted into soft one to do the different materials like slipper, bag, purse, like that, suitcase, like that. That process is called a tanning and curing method of leather. Then ethanol is used to make gasohol, that means is used as a fuel. Particularly in Brazil country, the ethanol is used as an fuel. It means eripurula பயன்படுத்துறாங்க. So these are all the different uses of alcoholic fermentation is the important 3-mark question. Dear students, see the general procedure of anaerobic respiration, that is glucose 6-carbon compound which is converted into 2 molecules of pyruvic acid by utilize the 2 molecules of ATP and NADH2. This pyruvic acid is involved in the 2 types of respiration. One is alcoholic fermentation, another one is the lactic acid fermentation. In alcoholic fermentation, 2 molecules of ethyl alcohol is produced and carbon dioxide is also released by the enzyme alcohol dehydrogenase in the reduction of NADH2 into NAD. In lactic acid fermentation, the NAD is also reduced into NADH2 is also reduced into NAD by the enzyme lactate dehydrogenase, producing 2 molecules of lactic acid, but there is no formation of carbon dioxide. So this is the overall process of anaerobic respiration. Next we are going to see lactic acid fermentation. It will be taken place in some bacteria, fungi and vertebrate mussels. Produce lactic acid from the pyruvic acid. See the equation, 2 molecules of pyruvic acid CH3CO COOH in the presence of NADH2 2 molecules by the enzyme lactate dehydrogenase produce lactic acid and reduction of NAD. That is said to be lactic acid fermentation is important 3-mark question. Write a short notes on lactic acid fermentation. Next one, mixed acid fermentation. It is a characteristic feature of Enterobacteriaceae and result in the formation of both the characteristic feature of alcoholic fermentation and lactic acid fermentation. That is formation of lactic acid, ethanol, formic acid and gases like carbon dioxide and hydrogen. That is said to be mixed acid fermentation. That means it is the process of both the fermentation types, alcoholic fermentation and lactic acid fermentation. Now we are going to see the characteristic features of anaerobic respiration. This anaerobic respiration is showing less efficient than the aerobic respiration. Number of ATP molecule is produced in this respiration is very less. This respiration is characterized by the production of carbon dioxide and it is used for carbon fixation in photosynthesis. So these are all the different characteristic features of anaerobic respiration. Is another important 3-mark question. Write the characteristic features of anaerobic respiration. Less efficient, first point. Second one, lesser number of ATP molecules are generated. Then production of carbon dioxide is used for carbon fixation in photosynthesis. Dear students, next important 3-mark question. Comparison between glycolysis and fermentation. In glycolysis, glucose is converted into pyruvic acid. Here in fermentation, pyruvic acid is converted into alcohol or lactic acid. Glycolysis is taken place in the presence of oxygen. It will be taken place in the absence of oxygen. 2 ATP molecules are produced at the end of the glycolysis. Here, the fermentation, no net gain of ATP is formation. 2 NADH2 molecules are produced, but here 2 NADH2 molecules are utilized, that means used in the anaerobic respiration. So this is the difference between glycolysis and fermentation. Is a very, very important 3-mark question. Dear students, this is the another table which are showing net product from 1 molecule of glucose under glycolysis and anaerobic respiration. Glycolysis, 2 molecules of ATP produced, reduction of 2 molecules of NAD, and total 8 molecules of ATPs are produced. In anaerobic respiration, 2 molecules of ATP produced and 2 reduced NAD is reoxidized. So only 2 ATP molecules are produced. So they given the star mark, that is 1 molecule of NAD get reduced into how many molecule of ATP? 3 ATP. Okay. So 2 3s are 6 molecule of ATP plus 2. So 6 plus 2, 8. Here NADH2 is not reduced, but it will be reoxidized so that it produces only 2 ATP molecules. That's why they given in the differentiate between, they produce more net gain of ATP in glycolysis, but in the anaerobic respiration there is no net gain of ATP formation is taken place. So this is the net product of glycolysis and anaerobic respiration. Dear students, next we are going to see important 5-mark question and a 3-mark question. Demonstration of alcoholic fermentation or anaerobic respiration. So this process was proved by a scientist named Kunz. So this fermentation is known as Kunz fermentation. In this experiment, the apparatus consists of a glass tube, contains a upright, that means straight glass tube with wide bulb. We have to pour 10% sugar solution inside the upright glass tube along with baker's yeast, and the mouth of the glass tube is plugged with the lid. After some times, the 10% sugar solution, that is glucose, reacted with yeast, which produce the alcoholic smell, and level of the glucose solution in the glass tube is reduced due to the accumulation of carbon dioxide gas. Due to the presence of enzyme zymase in the yeast, which converts glucose into alcohol and produce carbon dioxide. Now we can able to open the glass tube and add the KOH crystals. KOH is the good absorber of carbon dioxide, so it absorbs all the carbon dioxide gases which is produced in this reaction. Due to this reaction, the glucose level comes into the original position of the upright glass tube, indicates anaerobic respiration is taken place in the Kuhn's fermentation tube. Dear students, see the diagram. The diagram is also very much important for diagrammatic question. So this is the Kuhn's fermentation apparatus, a upright glass tube with a bend neck, end with a glass bulb with a lid. The entire apparatus filled with 10% sugar solution and baker's yeast. After the reaction taken place, they are produced alcoholic smell and the carbon dioxide gas, which is filled in the downward displacement of sugar solution and create the vacuum at the top of the glass tube. Now we have to open the lid and add the KOH crystals. The KOH is the absorber of carbon dioxide, so it absorbs all the carbon dioxide gas produced in this reaction. Once again, the sugar solution comes into their original position, indicates the anaerobic respiration, is proved by Kuhn's fermentation experiment. Dear students, up to this we have seen aerobic and anaerobic respiration and the different stages. Next, we are going to see the alternative path of glycolysis is known as pentose phosphate pathway or phosphogluconate pathway. So during respiration, that is anaerobic respiration, breakdown of a 6-carbon molecule of glucose is converted into 2 molecules of pyruvic acid in glycolysis process taken place in the cytoplasm or cytosol. So another pathway was observed by different scientists. It is an alternate source of glycolysis, is known as oxidative pentose phosphate pathway. Glycolysis is taken place out of 2%, but this pentose phosphate pathway out of 3, it will be 1%. This pathway was first discovered by Warburg, Dickens and Lippmann. So this pathway is known as Warburg, Dickens and Lippmann pathway. This is also taken place in the cytoplasm of a mature plant cell. This is an important 1 mark question. It is an alternative sources of breakdown of glucose. So another important 1 mark question. Dash pathway is an alternative source of breakdown of glucose. The answer is pentose phosphate pathway. So this pentose phosphate pathway, otherwise known as exose monophosphate shunt or HMP shunt, is the important 1 mark question. Or direct oxidative pathway. This process has taken place in 2 phases. One is the oxidative phase, another one is the non-oxidative phase. In oxidative phase, 6 molecule of carbon compound glucose 6-phosphate converted into 6 molecules of 5 carbon ribulose 5-phosphate with the loss of 6 molecule of carbon dioxide and generation of 12 NADPH2. See, in this oxidative phase, 6 molecules of 6 carbon glucose 6-phosphate converted into 6 molecules of 5 carbon compound of ribulose 5-phosphate and liberate 6 molecule of carbon dioxide and also produce the 12 molecule of NADPH2. Dear students, kindly note that one. This is not NADH, it is a NADPH2. So NADH2 is different from the NADPH2. The remaining reaction is taken place in the non-oxidative pathway, that is 6 molecule of 5 carbon ribulose 5-phosphate, which are produce different intermediate compounds, that is 5 carbon compound ribose 5-phosphate, xylulose 5-phosphate, glyceraldehyde 3-phosphate is a 3 carbon compound, pseudoheptulose 7-phosphate is a 7 carbon compound, and erythrose 4-phosphate is the 4 carbon compound. So these are all the different intermediate carbon compound: 5 carbon, 3 carbon, 7 carbon and 4 carbon. Finally, they produce 5 molecules of glucose 6-phosphate is regenerated in this pathway. Dear students, see the overall reaction of pentose phosphate pathway. 6 molecule of 6 carbon compound glucose 6-phosphate in the presence of NADPH2, that means 12 NADP plus 12 molecule of water produce 6 molecules of 5 carbon compound glucose 6-phosphate and release 6 molecule of carbon dioxide, inorganic phosphate. That means glucose 6-phosphate, 1 molecule of phosphate is liberated outside. That's why glucose 6-phosphate plus 12 NADPH2. So the complete oxidation of this pathway is 1 glucose 6-phosphate yield 6 molecule of carbon dioxide and 12 molecules of NADPH2. So the oxidative phase of pentose phosphate pathway is controlled by the enzyme glucose 6-phosphate dehydrogenase, which is inhibited means stop the high ratio of NADPH to NADP. Dear students, see the flow chart. Fate of glucose in HMP shunt and glycolysis. First one, starch converted into glucose via oxidation process of HMP pathway or pentose phosphate pathway converted into ribulose 5-phosphate. But in this glycolysis, by the oxidation process, it produces the pyruvic acid. So that is the two process which is taken place in glycolysis and HMP pathway. Dear students, this is the HMP shunt pathway or pentose phosphate pathway. The starting process is the 6-carbon molecule of glucose produce 6 molecules of 6-carbon compound glucose 6-phosphate by the enzyme exogenase. At the time, 1 molecule of ATP get reduced into ADP. So that process is called phosphorylation. Then the 6-phosphate, glucose 6-phosphate, which is converted into glucose 6-phosphate, which is a 6-carbon compound but 5 molecules. Then the glucose 6-phosphate is converted into 6-phosphogluconate, 6-phosphogluconolactone. It's a 6-carbon compound by the reduction of NADP into NADPH2. Then the next step, by absorbing the 6 molecule of water by the enzyme lactonase, converted into 6-phosphogluconate. Then again it will be converted into ribulose 5-phosphate by the reduction of another NADP into NADPH2. So here 6 molecule, then here 6 molecule. How NADP is produced? How many NADP is produced? 12 NADP is produced, and also 6 molecule of carbon dioxide is liberated. Then first step, oxidation. Second step, adding of water molecules, so hydration. The third process, oxidation and decarboxylation. That means NADPH2 oxidized into NADPH2. Then they release carbon dioxide molecule, that is decarboxylation. This ribulose 6-phosphate is produce different carbon compound of intermediate process, 3 carbon, 4 carbon, 5 carbon and 7 carbon. So that's why they given formation of phosphorylated compound. Then up to this, the oxidative phase is taken place. After that, non-oxidative phase. So the different intermediate compounds are converted into fructose 6-phosphate. Then fructose 6-phosphate again converted into glucose 6-phosphate. So this is the flow chart of pentose phosphate pathway or HMP shunt. Is the important 5-mark question. How they will ask the question means, draw a flow chart of HMP shunt or pentose phosphate pathway. So dear students, the last topic of this lesson is significance of pentose phosphate pathway. Is another important 3-mark and 5-mark question. First one, this HMP pathway is associated with the generation of 2 important products. One is NADPH2, another one is the pentose sugar, which are responsible for anabolic reaction. Another one, the coenzyme NADPH generated, which is used for reductive biosynthetic process and counter damaging of the effect of free oxygen radicals. Then ribose 5-phosphate is used for synthesis of DNA, RNA, ATP, NAD, FAD and coenzyme A. The 4-carbon compound erythrose phosphate is used for synthesis of anthocyanin pigment, lignin and other aromatic compound. So this is the importance of or significance of pentose phosphate pathway. So dear students, up to this the respiration lesson is over. Remaining we will see in the next video class. God bless you. All the best children.